Chapter 14, Mendel and the Gene. In this chapter, unlike other chapters, I will pick certain sections that I like and other sections that you do not have to know for the upcoming exam. For this chapter, we will look at Mendel's principles to see his experimental system, and we will focus on experiments with a single trait and the principle of segregation. Then we'll go on to the chromosome theory of inheritance, and we'll look at extension to Mendel's principles and human inheritance. So let's get started. Gregor Mendel is the father of genetics, and he always gives me hope because he was a man that had not succeeded in many areas in his life, and because of this he had ended up in a monastery. But in the monastery, he did some very prolific work with pea plants and started understanding how traits were passed from one generation to the next. And this is the chromosome theory of inheritance. And this was done through a series of experiments on garden peas, which is pretty fabulous. When we talk about heredity, what are we talking about? We're talking about the transmissions of traits from one generation to the next, or from parents to offspring. When we talk about a trait, we're actually talking about a protein that codes for a specific characteristic. Up until this time, people had some ideas about what was happening from one generation to the next. One idea was the blending inheritance, that offspring were a blend of somewhere in between mom and dad. Other scientists actually thought that you inherited traits that were acquired. Like you learned how to be a banker, so that was what you inherited from one generation to the next. And I think that would be awesome if I didn't have to go to school and I could just count on the education that my parents had already learned, so I wouldn't have to, that would be passed on to me and I didn't have to study it myself. But we know that that's not true. So the garden pea served as the first model for organism in genetics. Peas are inexpensive, they're easy to grow. You can grow many generations in 1 year. They produce a lot of seed, they're large and you can visually see them. And you can control which parents are going to have offspring. And these are characteristics which made peas a great model organism. The other thing is if you're messing around with which parents will be able to mate, no one's going to get angry when you're controlling reproduction of plants. Garden peas are very practical and a lot of information was gathered. The other thing that's important about pea plants is they have these things called polymorphic traits. In other words, there's several versions of the same trait, like different flower colors, seed color, seed shape, also pods. So they could look at there were a lot of visual things that you could see without actually knowing the genetics. Because remember, back in the day, actually Mendel and all of his colleagues, they knew nothing of DNA. So they had to go by on what they could see visually with their own two eyes. So peas can self-fertilize or self-pollinate, but you can also force them to cross-pollinate or have one plant share sperm with eggs from another plant. So you can control the mating. And the way that they did it is they were using brushes to spread pollen, and they still use this technique today. I have worked in places where they are very carefully controlling test crosses, and they'll use paintbrushes to actually spread pollen. So Mendel worked with pea varieties that had these traits that he could visually see: seed shape, seed color, pod shape, pod color, flower and pod position, and stem length. So all of these features that you can visually see, physically see, are called phenotypes. Phenotypes, PH, at the very beginning of the word reminds me of what you can physically see. So there are a lot of things that you can physically see in pea plants. Now, he worked with pure lines, hybrids, and true breeding lines. But for this class, you're not going to have to know this information. So let's skip it. Now, I do like this summary table 14.1. You need to know autosomal inheritance, gene, allele, genotype, phenotype, homozygous, and heterozygous. You also need to know dominant, recessive, and let's skip down a few, and maybe X-linked. So for sure, dominant and recessive on the top of this last part of the table. We're going to do a Punnett square, and if you are taking lab class, you know that we're doing them in lab. So on the outside of the Punnett square, you have the parental generation, and then inside of the Punnett square, the inside four squares are the offspring, or the F1, first filial generation. So Mendel crossed a lot of plants to see what kind of offspring he would have, and then he counted the traits. And so that's how he started learning how traits were passed on from one generation to the next. So here you see an example of some of the traits that he looked at, and these are for monohybrid crosses, and this is what you're responsible for in this class. Mono meaning one, so it is a one trait cross, and that's all we'll be doing in this class. So notice here in the first example, it's round seeds versus wrinkled seeds, and so let's take a look. He got round versus wrinkled, and he got this 3-to-1 actually ratio of offspring in the F1 generation. So he did it as a self-pollinated cross, and he also did it as an outcrossing, and he got this 3-to-1 ratio. When we have this 3-to-1 ratio, it indicates that we have dominant and recessive traits. If we look at simple Mendelian genetics, then if let's say we use big W, little w for wrinkled and non-wrinkled. So a wrinkled trait, so if it's round, it tends to be dominant. That means non-wrinkled. That means you only have to have one allele for it to be seen in the next generation. But for it to be wrinkled, you need to have two alleles, which makes it recessive. Dominant versus recessive. So the other way they could tell about dominant and recessive traits is by doing a reciprocal cross, but you don't have to know this for this class. So I love here that they have dominant and recessive phenotypes, and notice it's always a 3-to-1 ratio. There's 3 times more round seed that there are wrinkled, or 3 times more yellow seed than green, showing that round, yellow, and inflated pods are the dominant trait. And it goes on to flower color and stem length. Now, he came to this idea of particulate inheritance. He knew it was an acquired trait, and he knew that it wasn't blended, it was something else. Because remember, he didn't know about DNA and genes. So he called it particulate inheritance, like little package of determinants that actually acted as what was going to happen the next generation. So he would say they acted as discrete, unchanging particles. We know now that there are these things called genes and alleles. Okay, so remember what a gene is. It's just enough heredity or DNA material to code for a trait. And genes are found on DNA. So on a single strand of DNA, you can have many, many genes. When you put homologous pairs together, Remember, that's one from mom and one from dad. Then you have variations of a trait. My husband always tells me that my traits are very strong. Isn't he saying that they're dominant? His are recessive. So I only need one gene for mine to be expressed, but he needs two for his recessive traits. Okay, so each individual has two versions of a gene. This is often referred to as Mendelian genetics, and these variations are called alleles. One allele is found on mom, and the other one is found on dad. And remember, these are found on the homologous pairs. So that brings us to two more terms that you need to know, genotype and phenotype. Phenotype is what you physically see, and we've already talked about it. But genotype refers to genetic combinations, like big T, or is it big T, or two little t's. So if it's two big T's, it's homozygous dominant. If it's a big T and a little t, it's heterozygous. And if it's two little t's, it's homozygous recessive. So how is it that we get these ratios according to how the parents mate? And this is based on the principle of segregation. Remember in class when we were doing Punnett squares for the monohybrid crosses, and I drew sperm and egg on the outer part? That is, the pairs separate from the parents, and this actually happens in meiosis 1. So meiosis 1 is fueling this idea of the principle of segregation. And so this is how traits are transferred from one generation to the next. They separate into different gametes, like two different sperm or two different eggs. And so depending on which egg or which sperm is used for fertilization, that will be the trait that is represented in the next generation. So here are the terms homozygous and heterozygous. Notice here maybe big R is used for red and little r's may be used for white. So here we have homozygous dominant or homozygous recessive with the little r. And hetero means different. So heterozygous means two different alleles, big R, little r. So they talk about pure lines, but you don't need to know that for this next coming exam. But you do need to know homozygous and heterozygous. And you do need to know how to do a monohybrid cross. That is a cross for one trait. So here we see a Punnett square for a monohybrid cross. And so here it is, they're looking at big R is for round seed and little r is for a wrinkled seed. So notice that both parents are heterozygous, and when they mate, then we get a 1 to 2 to 1 ratio. And so this shows us dominant and recessive traits also. Okay, so this gives you some of his comments, Mendel's claims and comments of some of the offspring from dominant and recessive traits. Okay, so when we predict the offspring use the genotypes and phenotypes with the Punnett square, we have to create a table. And I must say that I will skip this because this is an activity that we did together when we were face to face. I will skip the dihybrid cross. We will only look at one trait at a time and not two. We will also skip the test cross. Okay, now let's go to 14.4, the chromosome theory of inheritance. Chromosome theory of inheritance rose from observations that Mendel saw and others like Sutton and Bovary, that genes are located on a chromosome, and that when we look at different traits, there's a principle of segregation, that the traits segregate out between different either eggs or sperm, and that there's a probability of what we will see for the next generation. So how is it that we can explain Mendel's principles? And then a lot of it goes back to the specifics of meiosis 1. Genes for different traits assort independently in meiosis 1 because they're located on different non-homologous chromosomes. So remember how they separate. The purpose of meiosis 1 was to separate homologous pairs. So depending on which pole they went to depends on where you see the trait. So this is called the principle of independent assortment, and this affects segregation. So here they're looking at different traits and seeing how they segregate depending on where you see them. This is known as the principle of segregation. Pairs of alleles are separated during meiosis 1 in the formation of gametes. So it depends on how they line up during metaphase 1. So this is critically important. So it shows you like exactly why we have this probability of a 1 to 2 to 1 ratio, depending on how actually the chromosomes separate. So we won't look at wild types or mutants, so I'm going to pass this up. And we know we have sex chromosomes. So it is important for you to know as students that 2 X's refer to a female, and an X and Y actually codes for a male. Now traits can be sex-linked, so you can have X-linked genes and you can have Y-linked genes. But the truth is that X-linked genes are way more common, and the reason for this is because the X chromosome is way larger than that of the Y. So you can have sex-linked traits, and these are traits that are found on the sex chromosomes. So what I'd like to talk about is, I would like to talk about inheritance, but I think I would like to focus more on non-sex chromosomes than sex. I think it is important to know that some are sex-linked, but it's also important to know about those that are not sex-linked. In other words, autosomal inheritance. Okay, so I'm going to skip this, and I'm going to skip this, and I'll skip this. And I'm going to keep on going, so I hope you can keep up with me. I'm skipping several slides. This will be skipped also. I'm skipping this. Okay, I'm skipping figure 14.14 also. Skip. Okay, now I'd like to talk about multiple alleles instead of just 2. When I say just 2, that would be one from mom and one from dad, or simple Mendelian genetics. But I think intuitively we know that there's way more genes that are affecting most of our traits than just 2. If we look at our eye color, even though maybe most people along the border have brown eyes, they definitely have different shades of brown. So what's controlling all of those different shades is multiple alleles. Another classic example of multiple alleles, more than 2, is our blood. So we have A blood, B blood, AB blood, and then O blood. So we have multiple alleles that is affecting our blood type. So that is important to know. The other thing that is also important to know, that instead of just dominant and recessive traits to look at phenotype, there's other forms. So there's another form of dominance called complete dominance. So this form of complete dominance is you have dominant and recessive. But you can also have codominance. So what does codominance mean? Well, codominance means that each trait is actually expressed, and that would be for blood types, like AB blood types. So each type is expressed. Or what about when you see dogs or even cows that have multiple colors of their fur? So those are codominant traits. I had a basset hound that was tricolored, had 3 colors. Those were all codominant colors. And then the last kind of comparison that we need to look at is incomplete dominance. It says alleles are not always dominant or recessive, they're incomplete dominance. And that means there's a blending of the two. So a red flower and a white flower will make a pink flower, a blending, and this is called incomplete dominance. So here's an example of that. So I want you to understand pleiotropy and the opposite of pleiotropy. So what does pleiotropic mean? So it says here some genes influence many traits. So it says these genes are said to be pleiotropic. So we can have a gene, one gene, and it can affect many traits. So one classic example is Marfan syndrome. But what is Marfan syndrome? Well, Abraham Lincoln was supposed to have Marfan syndrome. So this is a variation in one type of trait, and it makes for a very tall person that has like a very bony skull, and very long hands, and extremely tall. So notice one gene is affecting many traits, and that's the Marfan syndrome. Another one would be, I'm trying to think of another one, is hemophilia. With hemophilia, it's blood clotting, but also you become anemic, you have brittle bones. So it is one gene affecting many traits. It's pleiotropic. One gene is affecting many traits. I'm going to skip this, and I'm going to skip this. I'll skip this too. Okay, so the next thing that I'd like to talk about are traits determined exclusively by genes? And I think you intuitively know that the answer is maybe no, that environment can also affect how traits are expressed. I've often mentioned that I've had 3 sets of identical twins in my teaching experience, and I could actually tell the identical twins apart. And as you see identical twins get older and older, you usually see that they start looking different. Well, why is that if they have the exact same genetics? It's because they have different environmental effects, and these environmental effects affect the way that their genes are expressed. And the classic example they give in your book is PKU. You know, when babies are born and after they're born, you have to go back to the hospital and they prick the little baby's foot. Well, what they're doing is they're testing for PKU, whether you're homozygous recessive, for enzyme coding that gene. And this enzyme converts phenylalanine to tyrosine. And if it doesn't, and you have an accumulation of it, it can cause retardation. But if you recognize that it's not existing in the baby, then you can modify the diet. And once you modify the diet, which is an environmental influence, then you can have a normal individual. And that's why this testing is done. The next thing I'd like to talk about is quantitative traits. So quantitative traits is the opposite of pleiotropy or pleiotropic traits. Why is it the opposite? Because it's many alleles affecting one trait. So a classic example is human height. And how do you know that it's many alleles affecting one trait? Because if it were just tall people and short people, wouldn't people just be 2 heights? But instead, we're medium, tall, medium tall, medium short, all different kinds of heights. So we know that there are many alleles affecting height. And so usually quantitative traits means that many alleles are affecting a trait. And you know this because if you look at a population, you see a lot of variation forming this bell-shaped curve. So quantitative traits are affected by many alleles. Okay, so skin color, height, eye color, intelligence, all of these are affected by many traits, or many alleles, excuse me. These are quantitative traits. Okay, so we have one more summary table. I think you do need to know sex linkage, codominance, incomplete dominance, perhaps pleiotropy, polygenic, and environmental effects. I would like to talk about pedigrees because if you know if something's wrong in your family, then you should have a pedigree done, because when you have the next generation, you want to make sure that we can provide the best for them, and that also means the best genetically speaking. So we can analyze pedigrees in a different kinds of ways, but I think I would like to focus on autosomal traits. That means non-sex traits, those that are not linked to your sex chromosomes, XX and XY. Okay, so the first example is an autosomal recessive allele. So notice that it's a recessive trait, which is a small letter, and you need two alleles for the trait to be expressed, and autosomal means that it's on a non-sex chromosome. So usually what's happening in this case is that both of the parents, one or both of the parents are carriers, and they don't realize that they're carriers because usually they have a dominant trait, and that dominant allele is actually masking the recessive one. So they're a carrier, and oftentimes they don't know it because it's that trait is a recessive trait. So let's look at the pedigree, which you'll be responsible for. So if you look at the first generation, that's the Roman numeral 1, you see a square and a circle. The square represents a male and the circle represents a female. We know that they're carriers, in other words, that they have one recessive trait because it's half filled, it's half blue, it's a half square, and it's a half circle. So this man and woman have exactly how many children do they have? Yeah, you're right. They have 3 children. They have a boy and they have 2 girls. The boy, the male, has the trait. One daughter does not carry the trait, and the second daughter is a carrier. I hope you can see that. The male marries a female, and the female does not have the trait. But because the male is affected, they have 2 offspring in the 3rd generation. And notice those 2 children, one's a boy and one's a girl, and both of them are carriers. The 3rd daughter in generation 2 gets married to a man that's also a carrier, and they have 3 children, 2 daughters and a son. And a daughter and a son are unaffected, but they have one affected female because the circle is blue. So what are some of the traits of an autosomal recessive trait? Males and females are equally affected. Affected offspring often come from unaffected parents because the parents are carriers and they don't know it because it's a recessive trait and it doesn't show. You don't see the phenotype. And so affected offspring are homozygous. In fact, they're homozygous recessive. So if both parents are heterozygous, then there's a 1/4 or 25% chance that their offspring will be affected. And often this affects one trait and skips one generation, affects one generation and skips the next. So this is a pedigree of a recessive trait, and one classic example is the sickle cell disease. Now let's go to an autosomal dominant trait. I'm just reminding you that autosomal means a non-sex. So dominant, you only need one allele for it to be expressed. And the classic example is Huntington's disease. Huntington's disease is a terrible genetic disorder where you slowly lose your mind, and you usually find out after you have children. So after your reproductive years, you actually see that it will be expressed. Okay, so Huntington's disease. So it says here, homozygous or heterozygous individuals have the dominant phenotype. So remember, it only takes one allele for it to be expressed. And this will be passed on to about half the offspring. So let's take a look at this one. Let's see how this one differs from the other one. This is for a dominant trait, Huntington's disease. So notice that the female is affected and she only needs one allele for the trait to be expressed. She marries a man that's unaffected. They have 2 children, a male and a female, and they're both affected. These 2 children marry unaffected partners. And notice every generation, 50% are affected. So by generation 3, whether they have 2 children or 4 children, half are affected because it is a dominant trait. So what are the characteristics of a pedigree for an autosomal dominant trait? Males and females are equally likely to be affected. Affected offspring have at least one affected parent, and it's obvious. Affected offspring are heterozygous if only one parent is affected. So it only takes one allele for it to be expressed. And so about half the offspring will be affected. And this does not skip a generation. 50% of each generation will be affected. Now, the rest of the chapter talks about sex-linked traits, and it's good for those of you that are interested in genetics, but this will not be on the next exam. So I'm skipping a lot of this, and now we're at the end. So best of luck. This is the end of this chapter. Thank you.